Treatments for amyloids have centered around vaccine treatments, which the idea essentially is to create an antibody whose target antigen is one of these amyloid molecules. The most developed example of something like this would be the Alzheimer's vaccines. So the proposition basically is that Alzheimer's disease is caused by amyloid beta peptide. So the idea is therefore to vaccinate people against that amyloid. So what will happen is that the immune system will clear it out. And this has been done with some success. There was a trial in 2000 or 2001. It was halted because a subset of the patients who were vaccinated for this, 6% or so, had gotten a brain inflammation, which is basically a side effect of the immune system attacking too intensely the amyloid, and as a result the brain was damaged. But people have been working on different types of vaccinations, or many ways to vaccinate against something, or passive vaccinations that don't really involve a full inflammatory response. So there are other people working on it. Here's a look at some results of vaccines that have been tested recently in mice. So here are some genetically engineered mice that are designed to have this amyloid build up in their brain. In this study, let's see, experimental design. They had some mice that were divided up into three groups. One group, Group A, immunized with a cocktail of synthetic beta amyloid, S1P, and liposome. And this is the vaccinating agent, basically, and the other stuff is to help present this to cells that are responsible, that accept antigen, basically. So this is just to help it make it more effective. Group B consists of mice that are immunized with liposome alone. Not sure, quite honestly, why they did that. Probably just to see if... because this study also goes into the immune response as well, but I'm just not really going to go into that much detail because it's a long study. Probably to see if this causes any kind of immune reaction on its own, I suspect. C consists of something inoculated PBS, a control. Okay, so a control group, and we'll look just very quickly at just the amyloid results. So AB, amyloid beta. So here's a look at a slide of the brain tissue of one of these mice. Group B, so the ones that received only the whatever the L word was. Whatever that stuff was. Yeah, so you can see there's definitely amyloid accumulation. This is being stained for amyloid beta. Group C, same amount of staining as well, and then the control wild type. The wild type controls are mice that are not modified with this amyloid producing stuff, because the mice normally they don't get any kind of amyloidosis like this. So the only reason they get it in these examples is because they're genetically modified to produce it. And here are stains for different things: GFAP, CD45, CD3, so those are different immune cells, just to see what kind of immune reactions take place. So you can check it out if you want, but I'm just here to explain what the gist of what they're trying to do with these amyloids. So a beta amyloid is one, and also I thought I'd include here a study of that 2000-2001, I can't remember the year, but actually one of these vaccines made it into clinical trials of humans, and the plug was pulled relatively quickly because of this brain inflammation. It only occurred in, let me see, I think it was 6%, yeah, 18 of 298, 6% of patients, so not really that many, but still a very, very dangerous autoimmune disorder to live with, where the immune system is constantly attacking. It's like people who get organ transplants. they've got to take immunosuppressants because their immune system is constantly attacking that organ. It recognizes it as something foreign. And one thing I noticed in a study a while ago was the problem with some of these vaccines. They were vaccinating against this amyloid beta, and they were presenting it, but whenever they had a look at some tissue samples, they were noticing that not only there were more antibodies than were expected to be there, which means that once it began an attack against this amyloid, it started seeing other sections of it or other parts as it just created more antigens than they were expecting. So they're trying different methods, targeting different parts of the molecule, different types of vaccinations to try to mitigate against this inflammatory response. But of course, amyloid beta is not the only amyloid. It's just the most heavily studied. Alzheimer's research is a huge business, whereas the other ones, for a long time, were ignored mostly, and since they're very difficult to detect, and often they would only find them in the people of the most advanced stages weren't necessarily so concerned about, as I explained before, and it was difficult to tie to specific pathologies. But here is something very related. Here, this study, they're using radioimmunodetection of amyloid deposits in patients with AL amyloidosis. This is a very common amyloidosis. There are many amyloids. Most of them are not well understood, or just no one's really researching them that much. But here is one that is one of the more common ones that have caused problems to people, that people do look to get treated on a regular basis. So the significance of this is that here they're using radioimmunodetection. Basically what they're doing is they're taking one of these antibodies, and these antibodies are what is responsible for recognizing something as out of place in the body, something that is foreign. The body creates these antibodies, and these ends are antigen binding sites, and those are designed to bind onto things that shouldn't be there, and once they do, this handle, a cell will grab onto it and recognize it as something that needs to be removed. But what this study is doing, what these guys are doing, is they're taking advantage of this, being able to bind onto different surfaces. They're removing this part. This is the part where the immune cells grab onto it and recognize it as something that needs to get taken out. They get rid of this part and they put some other molecule that they can detect, normally something slightly radioactive, so when they do a scan, it shows up. But it stands to reason, I know it says only radioimmunodetection, but also if it works for radioimmunodetection, what they can do is just not change this for something that's going to show up on a scan and just leave it so that the immune system can remove it. So that's the importance of this. It's not really so much the radio detection, of course, that is very useful, but we're more concerned with the removal. And if it can be detected, if it binds onto there, then you can change the end of it and have it to create an immune response. And besides the potential side effects of an immune response, the only other problem that could possibly occur is once the immune system does respond to it, if it is unable to break it down. There's one thing that people have noticed about these different amyloids, Alzheimer's, all these amyloids included, is generally the immune system does attack them. Often they will find macrophages in the brain, I think it's microglia is what they're called, the macrophages of the brain. Whenever they look at these amyloids, they notice immune cells by them attempting to get rid of this stuff, and for whatever reason, A, they just cannot get rid of it fast enough, or B, what this study is going on about is that they are taking this stuff in, but they're just not able to degrade it, which doesn't help at all if the immune system attacks it and is not able to remove it. So what can be solved here is similar to the problem with lysosomes, where we had something similar to here. Instead of outside of the cell, we had inside of the cell that we went over with lysosomes. And that can be taken care of by getting the appropriate enzymes to the cells. In this case, the lysosomes of the macrophages. If we're able to get the appropriate enzymes to degrade this fibrillar insulate amyloid, which is an amyloid that has been recently thought to be responsible for type 2 diabetes. But what it's thought to occur is that there is an amyloid buildup near insulin-producing cells of this. And what will happen is that it will kill off those cells, and as a result, they're not able to produce insulin, and they become diabetic. So being able to treat this would be important, and for a vaccine to work against this, it's going to have to respond to it appropriately, but at the same time, it's also going to have to be able to get rid of it once there is an immune response. So that's where, or these are some of the bigger ideas that have been going on with removal of amyloids. Of course, there are other things. People have, for a long time, been looking at different molecules that'll break them apart. But still, you have stuff there, and if you break it into a bunch of pieces, it's not really clear that they'll go away just because of that. Of course, people have also been looking at stopping the enzymes responsible for producing this amyloid, but of course that will also have side effects. So out of all the things that are out there right now, this seems to be the most promising. And even though there have been some minor setbacks with the side effects of autoimmune disorders in some people, generally the people who have or animals or whatever they have tested it on, who have responded well to it, it has worked and it has done exactly what it says it was supposed to do. But people are, as again I said, are working on now just dampening down this immune response somehow. Once they figure that out, what will probably happen is they'll figure it out in Alzheimer's first, which is good, because if they figure it out in Alzheimer's, then for beta amyloid that is, and then when they apply, it'll be easy to apply to different areas, because the brain is such a sensitive organ. It's one of the more delicate ones. If they're able to get that to work, then the other ones won't be such a problem. In fact, maybe even techniques they're using now will work in other areas of the body where the possible collateral damage either A, won't occur, or B, just not be something as important because it regenerates well, or or for whatever reason it just doesn't, the damage is not as intense. And of course, the brain as well is the most important organ when dealing with these kinds of regenerative medicine sort of things, at least as far as lifespan is concerned, because that stores the person's personality. And in all honesty, if it really, really came down to it, you could probably do a whole body transplant, assuming that you could preserve the brain. And I understand that sounds a bit like science fiction, but it's not. It's already been done. It was done 40 years ago. Not in humans, but in chimpanzees. And if someone can do it in a chimpanzee, they can do it in a person. So that's pretty much it for this. If there are any updates on the vaccines, or perhaps some new ideas, I've had some interesting thoughts on other techniques that may be viable for removal of amyloids, assuming that you can get to them. In some places they're too in between the tissues, and it's not really that easy to get to. But I had some ideas for the tissues in the brain that are surrounded by that liquid, the cerebrospinal fluid. If I have any ideas, I'll put them on here. But that's it for now.